DNA is the molecule that defines each and every one of us. It is smaller than the eye can see, but it is found in each and every cell in our body. Everyone's heard that before, but what exactly is DNA and how does it work? That's what we'll be learning today in science with Max, Andrew, and Stephen.
This is a DNA molecule. DNA stands for deoxyribonucleic acid. It is formed in the shape of a double helix, essentially a ladder that twists and forms a spiral staircase.
If you unwind all the DNA in your body and stretch it out, it would reach the sun and back 4 times. A single strand of DNA is actually 6 feet alone.
The DNA molecule is a polynucleotide, which means it consists of many bonded nucleotides. The DNA molecule is made up of 2 strands that are composed of a sugar known as deoxyribose and a phosphate. These form long chains and create the outside structure of the molecule. The 2 strands are bonded with nitrogen bases and nucleobases, specifically adenine, thymine, cytosine, and guanine.
These 4 chemicals use hydrogen bonds to connect their sugar phosphate strands. However, certain nucleotides can only bond with other specific nucleobases. Adenine is complementary to thymine, meaning it can only bond with thymine, making them complementary base pairs. Meanwhile, cytosine and guanine are complementary base pairs. These are base pairing rules, stating that certain bases can only bond with each other. The different base pairing create different codes, or a gene, which is part of the DNA strand that codes for a specific trait, like eye color or hair color.
However, DNA reproduces in a very interesting way, called DNA replication. DNA replication, in simple terms, is the process by which DNA reproduces, forming 2 identical DNA molecules. The process starts when an enzyme known as a DNA helicase breaks hydrogen bonds of complementary bases. The first step in DNA replication starts unwinding the double helix into 2 separate strands using the DNA helicase. The 2 strands are the lagging strand and the leading strand. The leading strand is the strand that works in the direction of the replication fork, while the lagging strand works away from the replication fork.
The replication fork is the Y shape created when the 2 strands separate from the original DNA.
The leading strand has a short piece of RNA called primer, which binds to the end of it. A primer is a starting point of DNA replication. From here, the DNA polymerase, an enzyme in the cells, binds to the leading strands and moves along the strand, attaching complementary nucleotides to the DNA. The nucleotides talked about before, cytosine, thymine, adenine, and guanine, are found in the cell floating about. This process of replication is known as continuous replication.
The lagging strand, on the other hand, has many different RNA primers on it, which are created by primase, another enzyme in the cell. The RNA primers bind on the DNA molecule just as it does with the leading strand. Chunks of DNA known as the Okazaki fragments, named for the 2 Okazaki brothers, are added to the lagging strand. These fragments face away from the replication fork. The fragments also essentially just form in the strand using particles from the cell and its surroundings.
After DNA is done replicating, the polymerase will go back over the newly formed DNA molecule, essentially proofreading for any mismatched nucleotides or any errors in replication. This reduces the chance of mutation.
As the DNA is replicating, it is also uncoiling from the chromosome, the X-shaped figure that contained the coiled DNA. As new strands are formed, the DNA coils back up, forming new chromosomes. There are always 23 pairs of these chromosomes in human cells, making 46 in total.
There are thousands of genes in each chromosome, which are defined by the DNA that is produced. Each time the DNA replicates, it should be exactly the same as the starting or original strand, but sometimes genetic mutations occur. Genetic mutations are when something goes wrong in the base pairing and the polymerase doesn't catch the mistake. Sometimes the bases can change in the process known as tautomerism, when a hydrogen atom will move on the base, changing the actual nitrogen base itself. This will cause an alteration in the DNA, which might cause something different to happen. Despite how it may sound, this isn't necessarily a bad thing.
Genetic mutation is how evolution occurred. Somewhere along the line, when an organism was reproducing, the DNA didn't replicate properly and it resulted in a mutation. This mutation may have been beneficial to the organism, and they passed the mutation along, resulting in new species and causing evolution. If it weren't for genetic mutation, people wouldn't have different eye colors, or even be human for that matter.
All in all, DNA, while being one of the most researched topics in all of biology, is still a key factor in figuring out who we are and is still largely a mystery. But without it, there would be no life. Thanks for watching.